Thank you, Mark. And I hope that you are seeing my screen here. So sorry, I'm just trying to get it to. Okay, there we go. Now I'm all happy. You can hear me okay?
All good.
Yes, okay. All right, so I am going to kind of walk us through the basics of gene editing, and I'm going to assume that there are some people in the audience that don't really know too much about it at all, and so I'm going to really go back to the basics of animal breeding, and that is that we can really select animals to give us a particular characteristics or appearance or phenotype, we call it in genetics, based on just naturally occurring genetic variation that exists in the within the DNA of different individuals. And so all of these dog breeds have been selected from the wolf just based on naturally occurring insertions, deletions, indels, SNPs, and to give them particular characteristics. And of course, breeders do this not only with our pet species, but also with our agricultural species. And one of the tools that has been used to do that is tools that include biotechnology. And so specifically, I think this group is well familiar with the long history of genetic engineering, or what has been kind of colloquially known as GMOs. And that really was things where we're introducing typically a transgenic construct to give our plants or animals a particular characteristic that is of interest to us. And you can see here that that started in animals back in 1985. So around about the same time that plants started with genetic engineering. And we can see perhaps in this little montage that I'm showing some familiar faces from the hits, the greatest hits of animal biotechnology, I guess. And so we had the beginning of genetic engineering, and then you may recognize here, for example, the Aqua Advantage salmon, which was the first approved genetically engineered food animal to come to market, and that was first produced back in the late 1990s. And then of course here's Dolly the sheep, who's an important part of all of this, because cloning is a really important reproductive technology to use in the production of genetically engineered animals. And then what happened here in the kind of the middle of the early 2000s was we started to have the introduction of these things called gene editors, zinc fingers, talons, and CRISPR-Cas9. And that's going to be a lot of the focus of today, because we're kind of entering a new era, moving on from random integration of genetic engineering constructs to targeted alterations at particular locations in the genome, and that's known as gene editing. So I'm going to go to the next slide and say, well, what exactly is gene editing? And basically, it's broken down on this slide, and there's a couple of different editors, and what their names are is kind of unimportant. Well, zinc finger nucleases, talons, CRISPR-Cas9, and they all do the same thing as their name suggests. They make a double-stranded cut, nucleases. They cut the DNA nucleases, and how they do that and where they do that is guided by the zinc fingers or the talons or CRISPR-Cas9. And once this double-stranded break has been introduced, the cell tries to repair that double-stranded break, and it can either be done, and I'm going to call this the left-hand pathway here, and that's basically by a thing called non-homologous end joining, and it just sticks the two ends back together. And sometimes you'll get a small deletion or a small insertion that, if it happens in the reading frame where the three nucleotides are being converted to amino acid, can throw it out of frame and introduce an early stop codon and inactivate or knock out the gene. So that's one pathway. That's kind of the inactivation pathway. I call that the left-hand side. And then the right-hand side over here is basically providing a template that would then integrate using homology-directed repair that enables the introduction, in this case, of this pale blue DNA at the location. And those is, I call this the right-hand pathway. So they have a repair template, and you can do things like introduce an allele from maybe one breed to another, or you could technically introduce foreign DNA like a transgene if you wanted to do that. And so I'm just going to hopefully share here. I just need to do something to enable the sound here. And so I'm going to stop sharing for 1 minute so that I can now do this sharing. And I apologize for this. It was not allowing me to share sound a minute ago. So here we go. And now I'm going to play you a little video that just basically outlines what I just said.
For centuries, animal breeders have selected individuals displaying desirable qualities and bred them to produce particular goals. Breeders are actually selecting on naturally occurring DNA sequence variations that result in differences between individuals, like the color of a cow's coat. Using conventional breeding programs, achieving desired changes can take decades, especially in large livestock species like cattle, which have long generation intervals. Innovations in biotechnology are poised to help breeders improve the rate of genetic gain. Genomes are made up of DNA, containing the collection of genes, unique instructions detailing every characteristic of an individual. What they look like, the amount of milk produced, or resistance to disease. Genome editing offers an opportunity to edit these instructions precisely. One method to edit genomes is the CRISPR-Cas9 system. This system has two components: the Cas9 protein, which cuts DNA, and a guide molecule that directs Cas9 where to cut. Bound together, they form a complex that introduces a precise cut in the DNA. The break is then mended by the cell's natural DNA repair machinery. This can be used to inactivate undesired genes, like those that cause disease. Alternatively, the repair machinery can introduce useful DNA sequences which result in desirable traits, like heat tolerance. Genome editing complements traditional breeding methods. It has the potential to introduce useful traits like disease resistance and adaptability into genetic improvement programs, addressing some of the problems facing global agriculture.
So I very deliberately showed you that video, which is available for free on YouTube, just as a simple introduction to the topic, because I think sometimes it gets so complicated in technical terms that we kind of lose the general public. And so anyone that ever wants to show that video, it's available to you there. So how do you go about genome editing a cow, for example? And it's a little bit different to plant transformation, although there are some similarities. And basically what's important is we need to introduce this useful variation, whether it's a knockout or a knock-in, into cells that are ultimately going to produce gametes, that is sperm and egg, in the resulting animal, so that it can be passed on from generation to generation. Because as animal breeders, that's our goal, is to actually pass those genetics on. And so we're interested in germline editing, somatic cell editing, although that's important for human therapeutic purposes, is really not our objective in animal breeding. And basically what's shown here is a one-cell zygote, so just after the sperm has fertilized the egg, so there's one cell in there, and you can introduce your editing reagents into that cell, and ultimately what we want is this animal that has the edit in both alleles, typically, is what we want. And it can be done in basically two ways when we're talking about mammals. The first way is basically doing the cell editing in somatic cells, and then taking an egg from a donor female, removing the nucleus, and then basically taking that 2N nucleus that's been edited and putting it into an enucleated oocyte and activating it, and basically doing what's called cloning, or somatic cell nuclear transfer cloning. That's why Dolly becomes important again. And so then that gets gestated in the case of a cow for 9 months in a surrogate and gives birth to a homozygous non-mosaic offspring. So that's our holy grail right there. And that has some advantages in that you know that this animal is going to have all of its cells be the same because it came from a single cell. And you know what you're transferring basically, and you can do editing, knocking in pretty easily and replicating cells like this. But cloning is pretty low efficiency, and not all cell lines clone well, and some countries don't allow cloning. So the other approach that's used is something where we take the female's egg, we introduce the editing reagents following fertilization with sperm in the zygote or the embryo stage, and then we put it into a surrogate cow. So that ends the same. And if we get very lucky, we might get a homozygous non-mosaic animal if the edit occurs in the one cell stage, or maybe a heterozygous animal. But more typically, what we get is something called mosaicism, where we'll have some cells that have the edit and some cells that don't, because it wasn't introduced at that one cell stage. And then we'll need to breed through to get to our homozygous non-mosaic animal. And if you're interested in this, I've got citations for everything I talk about, and you can read more. These are really overview slides to give you a feel for what we're doing here. So this paper goes through genome editing approaches to bring edits into breeding programs, and I would recommend it if that's something that you're kind of just getting up to speed with. If you've always worked in plants, for example, and now suddenly you're being asked to look at cows, it'll give you some background information. So what I just mentioned was cows, and it's going to vary dramatically depending if you're working with a mammal like a cow or a sheep, and a chicken, for example, like my colleague Mark does. And there, the editing is done totally differently, because when an egg is laid, it's multicellular, so there's no way to kind of get to the egg at that stage. And so typically in chickens, for example, we're working to basically take what's called primordial germ cells from the gonads and purifying them and then doing the editing in these cell lines, which are then brought back into an egg of a developing chicken to produce the edited animal. And there is a section on chickens tomorrow, and you'll go into more details here. I just bring this up because I want to emphasize there's going to be dramatically different approaches used depending upon the species that we're working in. And so cloning doesn't really make sense in chicken language. And similarly, you can't do what I just outlined in chickens in mammals. So it'll depend on whether you're a fish or a mammal or a chicken as to how that might happen. I did want to just go into a bit of nerdy classification here, and if you want to read more, I've got the citation at the bottom, but you'll often hear us use the term SDN1, and that's basically the left-hand pathway I outlined earlier, where we're introducing an edit, but not introducing any template. And so there's no foreign DNA that comes into the animal. So we're typically used to inactivate a gene, not always, but typically. And that's called an SDN1. So you've got the non-homologous end joining. SDN2s are using editing, but then providing a template that's usually a short template to enable the insertion or mutation at the target site. And you want to maybe particularly introduce one nucleic acid that will alter an amino acid, for example. So small mutations typically is an SDN2, and SDN3 usually involves a long template, and that would be used to maybe introduce an entire gene, for example. Maybe it's the same species, maybe it's a different species. But I wanted to define these terms because I think they're important to understand. So SDN1s or knockouts are things that we might use to knock out, for example, genes associated with allergens that make people allergic to milk, for example, or we might use it to inactivate a gene that gives an animal thermotolerance, and we'll hear that example given in the cow section with things like the SLIC mutation. We can do a sex ratio skew, so produce all female pigs, for example, so we don't need to castrate males because we've inactivated a gene that results in male development like SRY. We can stop unwanted development like boar taint, increase yield through things like myostatin, and then we have things like disease susceptibility, which is obviously something that's very important, and then surrogate germline and brood stock. Okay, I do see a hand raised there. Maybe Chantel, can you put your question into the chat so that people can see what the concern is? Thank you. Alternatively, using a template, so an SDN2 or 3, we might do actually similar things. So we're not really changing our breeding objectives because we're using editing. We're just altering the way that we achieve it. So we might produce animals that are susceptible to disease, have unwanted development, thermotolerance, things like improved food quality, and again, sex ratio skew. And I think we'll hear an example where researchers are trying to produce all female chickens for the layer industry. So when we did a review of all of the publications on genome editing in livestock for food purposes, this is a summary of what we found. And so what you'll see is about 80% of all of the edits are being done using CRISPR-Cas9, or at least this is publicized, as in published papers in the peer-reviewed literature. And if you want to read more, it's in this EFSA supporting publication that's available freely. And when we broke it down a little bit more, about 30% of the editing was done in cell lines, followed by cloning. So obviously that's in mammals. So 30% were cloned. About 60% were done in developing embryos. So that would be, for example, microinjection and introducing the editing reagents in a zygote. And in the case, obviously, of mammals, you have to go back into the surrogate animal to put those back. And then other approaches is going to be things like chickens, where you're doing that crazy primordial germ cell thing and going back into the egg. And that's basically what the overarching use methods was. And then about 75% of all applications that were in the peer-reviewed literature are SDN1s. That is knockouts that are not basically introducing any novel DNA. So there was a smaller percentage of SDN2s and SDN3s. And depending what you want to do, each has its utility. So why are researchers doing this? What was the purpose listed on the paper as to why they were trying to edit these animals? And this broke it down into these different topics. So abiotic is things like resistance to high temperature, so like the slick allele. Biotic stress is basically making animals disease resistant to things like PRRS or tuberculosis. Color is important for some of our aquatic species. Hypoallergenic is knocking out an allergen. Multiple is as it sounds, so not just one trait, but we're doing multiple traits, so stacking the traits. Reproductive characteristics, things like single sex offspring, that type of thing. Altered meat quality to improve the quality of the consumer's experience. Yield, and then other traits like welfare, introducing traits like hornlessness so that animals don't need to be dehorned to protect themselves and their human handlers. And this breakdown, which is in a recent publication, is shown here, is kind of the preponderance or the breakdown of what has been done in which animals. And so what you see in blue, ruminants, is cows and sheep. Purple is monogastric mammals. Aquatic is orange there. It's fish. And then chickens or poultry is in yellow. And so you can see yield is number one. Reproduction, number two, and biotic stress, so disease resistance, is number three. And you'll see that's a really important focus that people are going towards. If you're interested, you can go and have a look at this paper. We've also put together a website called the Gene Edited Animal Database. EJ from ISA has been responsible for this, and we'll talk about this on the last day of the session. But if you're interested, you can go to this link and find all of the different papers that are there. And if you go into it, you'll see there's a filter where you can filter, for example, by species or what trait categories, what genes. And so in this example, I'm going to click, this is all of the different species that are in the species link. And if I, for example, click on sheep, I'll get a breakdown of what papers were done in sheep, what year they were published, who published them, what the gene target was. And if I click on view details, I can go and get more details on that paper. And so this is an easy way for you to get up to date on what's going on in terms of in terms of this field. And there's an important button up here that says no. That means that these are not products that are going to commercialization at this stage. This is just peer-reviewed papers. It doesn't mean that they're necessarily getting commercialized. So this undergone regulatory process, no, will give you that breakdown. And that's important because regulatory, as we'll hear later on, is a really important step in commercialization. And so where are these animals being produced. And this is kind of a breakdown of the different countries that are the primary author of the peer-reviewed publications that we reviewed in this ESFA publication. And you can see the breakdown here with China being a very dominant player in this particular field, but you can have a look at your particular country there. And then I think you can also look in the gene edited animal database and have a look at your particular country and at what is happening, if anything, in terms of gene edited animals there. And I will just draw your attention to this. This has undergone a regulatory process. So I've changed the button here to yes. And this is basically a breakdown of which countries have evaluated, or at least have the regulators in that country have looked at a gene edited animal application and have made a determination regarding that application. And if you click on here, you can go to view details and it'll tell you where. So this is different years. And so for example, here's Argentina back in 2018, evaluating a Nile tilapia that was a myostatin knockout, and you can view details there. And so that gives you actually everything that has gone through a regulatory process to date is listed on this slide right here. And so that is also some background information for regulators that I think might be useful. And that's, again, undergone regulatory process, yes. And so you'll see here, Japan has a number of animals that have gone through a regulatory process. And actually, in this case, has an example of 3 fish that have been produced, actually by an academic researcher, I understand, started their own company to produce fast-growing fish that are either knocked out for myostatin or the leptin receptor. And so you can see here brim, puffer, and flounder fish, and they've been evaluated and have been given permission to go to market in Japan. And so I just hold that up as an example of there are actually products that are coming to market as we speak. So I hope that's given you an overview that genome editing offers an approach to introduce useful genetic variation and alleles for large effect loci. And we'll hear more about disease resistance as an example. So the porcine reproductive and respiratory syndrome virus is fast approaching market, and that's a pig disease that we'll hear about. And welfare traits like not growing horns into cattle breeding programs. The way they're introduced will differ by species, dependent on the biology of that species. And so it's quite different to plants, and it depends on the species as to which method is going to be used. And that will potentially affect some aspects of, for example, a risk determination for that particular species. When we looked at all of the peer-reviewed publications, there's about 30% where editing was done followed by cloning, which will have different implications in different countries. There was about 61% that were done in developing embryos, and then mostly chickens were done using other approaches in primordial germ cells. And if you want to look at the different types of edits, you can go to the database that's listed here again. But CRISPR-Cas9 was about 80% of the peer-reviewed literature, and SD1 edits was about 75%. So knockouts using CRISPR-Cas9 is by far, at the current time, the most common edit that we're seeing in the peer-reviewed literature, which is usually a bit of a view to the future as to what's likely to be coming down the pipe. And so I encourage you to check out that database, and I just want to acknowledge the groups that fund my lab. And at this stage, I will stop sharing and pass the microphone back to Mark.